Good morning. I'm David Esposito, a little short compared to the others, all right? I want to thank the organizers of OIS for the opportunity to present and give a quick update on ONL Therapeutics. ONL, we're based in Ann Arbor, Michigan, right outside the university where our technology first started. About 18 months ago, we raised a Series B round, close to $50 million. BIOS Partners out of Dallas-Fort Worth was a lead investor. We've had a number of investors in the crowd here today, the ophthalmology-focused VC firms, Excite, and also InFocus Capital Partners have been supportive of us. We also have some strategic investment in both Janssen and Novartis have been very helpful in our development along the pipeline. What we're focused in on is developing therapeutics to inhibit the FAST receptor. Our lead compound, ONL-1204, is right now active in 3 phase 1 programs of distinct indications, one being retinal detachment, second in GA, and then the third in open-angle glaucoma. So far, the data in those phase 1 programs look really good, and we're now looking forward to raising a Series C round to be able to fund those phase 2 programs across those 3 indications. A little background on the technology. You saw some of the mechanism of action a little bit earlier. We feel with FAST receptor as a cell surface receptor on the surface of retinal cells. We see it as upstream of a lot of the other targets in the market, and we feel very differentiated by blocking upstream at the cell surface effect. We, at the time of injury with the activation of FAST, triggers a death signaling pathway of that retinal cell, also triggers the inflammatory cascade. ONL-1204 targets blocking that FAST receptor across a variety of disease states in our preclinical models. And what we really discovered over the last 12 months in our study in retinal detachment is that we've now confirmed biological activity in humans of ONL-1204 blocking the FAST receptor. Our study in retinal detachment, it's a surgical procedure, so we have the opportunity to assess human vitreous and the impact of our drug. We're clearly seeing reductions in the cytokine inflammatory environment in human vitreous, and that's given us a great deal of confidence as we move our programs forward that we are blocking upstream of other potential pathways to be able to protect retinal cells, but then also quiet the inflammatory response across a number of disease states. We've made steady progress in our pipeline over the last few years. Highlighted here are what we see as an initial focus on acute indications of retinal detachment. We're just about to conclude that phase 1 study and move into our phase 2 program. We also see opportunities collectively in an acute portfolio with potential other indications of acute glaucoma. We've made good progress. I'll talk to a little bit on the specifics on AMD here, but in our dry AMD study, in addition to open-angle glaucoma. We've also made good progress on our gene therapy program, which is basically using an AAV2 vector to transduce ganglion cells and Mueller cells to secrete soluble FAST ligand, which acts as a similar agonist as our peptide on the FAST receptor. So good progress, a little bit of details on the update. So retinal detachment indication, this was a dose escalation study, 4 doses. All 4 doses of ONL-1204 were safe and well tolerated. As I mentioned earlier, we've confirmed biological activity in humans on the FAST receptor, which we feel again gives us good confidence going forward with the program. But also importantly, on top of standard of care surgery, we're seeing with ONL-1204 on board, best corrected visual acuity improvements at 6 months, which is above those natural history trends. So we feel real confident about moving that program forward into our phase 2 program. In terms of AMD, I'll show you a schematic in a second on the study. But right now, so far, with about 10 patients in, the data looks well, safe, and well tolerated. We're also assessing, obviously, the visual assessments on progression of lesion size, but we're also seeing good functional improvements already in the study, and we're excited about that. In open-angle glaucoma, we have, again, about 10 patients in that study. So far, so good, and we hope to be reporting on that coming forward. And in our gene therapy program, we are looking to start a non-human primate study early next year. So good, steady progress across the portfolio. Specific to this conference and then with AMD, this is our study design of our phase 1B study. We're looking for 6 months of a natural history. This is a 30-patient study. We're looking at the natural history of the disease. We're also using a number of different exploratory biomarkers. We're doing aqueous taps to be able to assess the inflammatory markers. We're also using the innovative technology out of Novi and Francesca Cordero's lab with a DARK technology that's able to image sick and dying retinal cells. We're doing that periodically in the natural history phase. We're also doing it in the treatment phase. So we're now randomizing and treating for the next 6 months with ONL 1204 at a high dose, a low dose, and a sham. And one important to note, we feel the durability of our product throughout our preclinical work and what we're seeing in the clinic supports at least every 90-day dosing. We think we could potentially go out to 120 days. We've just started the treatment phase of that, but what's highlighted in that yellow box is we had an open label dose escalation arm of that study that we just completed. 3 doses, single injection with ONL 1204 in GA patients. Not only are we seeing safety and tolerability, but we are seeing initial good functional benefit in terms of letters read. And so we'll have to continue to progress with the study, but so far so good with our initial aspects with regards to our AMD program. As we prepare now for the next chapter of the company, our next clinical programs in phase 2, we've been guided by a scientific advisory board that helps support this. The vast majority of the retina community have been very supportive of our progress in the acute indication of retinal detachment. In addition, our board and our investors, BIOS Partners, is committed to leading or co-leading our C round. This is the initial part of the C round, a $20 million round to be able to fund the phase 2 retinal detachment program. It's about a $500 million US market opportunity. We're excited about that, but we also see the potential for a much larger C round as we progress into phase 2 programs on AMD and also open angle glaucoma in addition to our gene therapy. We're super excited about the future. We see real potential with fast inhibition, given its unique mechanism of action across not only the large chronic indications that we've been talking about, we also see some very unique unmet needs in the acute indications as well. And we look forward to updating you on our progress in the months and years to come. Thank you very much.